Now that we've discussed how the action potential occurs via ions running in and out of the axon, we now need to talk about neuroglia, because one type of glial cell in particular, oligodendrocytes, is going to assist in this process of an action potential. It's going to help generate something called saltatory conduction, which we'll talk about shortly. But first, let's just talk about the three general types of neuroglia. Neuroglia means glue, or glia means glue, and neuroglia means brain glue or neuron glue. And that's because when these neurons were first discovered, not neurons, when these glial cells were first discovered, they were thought to simply hold neurons in place. Well, it turns out that they do in fact do that, particularly astrocytes help kind of glue neurons together, so to speak. But they do more than that, and there are more types of neuroglia than just astrocytes. We have astrocytes, oligodendrocytes, and microglia. Astrocytes, astro meaning star, cyte meaning cell, astrocytes help form the blood-brain barrier. They help bring nutrients to the neurons, and they're involved in phagocytosis, which is a matter of getting dead material out of the brain. For instance, if a neuron dies, that neuron needs to be flushed out of the brain, and this is done through phagocytosis. The second type of neuroglia, the kind that gives rise to saltatory conduction, is oligodendrocytes. And oligodendrocytes have these arms that extend out from them that wrap themselves around the axons of the neuron. We can see this depicted in image number 2 on the left. These myelin sheaths, these arms of fat, wrap themselves many times around an axon, creating a sort of insulation, much like the insulation you would get around an electrical wire. However, there are gaps in between these myelin sheaths, and these are referred to as nodes of Ranvier. Ranvier being some French guy that discovered them. Again, when we talk about saltatory conduction, we will discuss how this is facilitated by the myelin sheaths and the nodes of Ranvier. And then the third type of glial cell is referred to as microglia. These also produce or perform the process of phagocytosis, and they additionally are involved in immune function. They protect the brain from invading organisms, and they're involved in the inflammatory response that is seen after brain damage. So 3 types of glial cells, and the one we're really going to focus on is number 2. As we said, oligodendrocytes generate these myelin sheaths, and the space in between them is nodes of Ranvier. So if we look over here on the left, we see the cell body of a neuron. We can see the dendrites out here, and then we have an axon extending out from the cell body. And we'll follow this axon collateral, and we can see a myelin sheath wrapped around it. Here is an oligodendrocyte cell body and the myelin sheath that is extending out from it. Again, then we have a node of Ranvier, and here a gap. Then we have our next oligodendrocyte generating a myelin sheath, and then we see a node of Ranvier in between it and the next myelin sheath. So this happens down the axon: myelin sheath, node of Ranvier, myelin sheath, node of Ranvier, myelin sheath, etc., until you get almost to the axon terminal, and that's where they stop. Over here on the right, we can see a photomicrograph, a cross section of an axon. The lighter gray area here is the axon itself. If we look up here and see what kind of looks like the head of a goose or something, we see an oligodendrocyte cell body with its nucleus in the center. And extending out from that oligodendrocyte is this myelin sheath, and we can see that there are many layers wrapped around again and again around the axon to give it a nice tight insulation. I'm going to skip these two laws of action potential for a moment and first explain saltatory conduction, then I'll come back to those two laws. Saltatory conduction refers to the process of jumping or dancing. And as we see down here at the bottom, we have an axon that's myelinated, and in those axons we get the rapid conduction of the action potential through the myelinated segment, but then it slows down at a node of Ranvier where the action potential is going to recharge. And then it'll go rapidly through the next myelinated segment and then slow down. Rapid, slow, rapid, slow, etc. until it gets to the axon terminal. Now this fast and slow and fast and slow repeated pattern is what is referred to as saltatory conduction. It's as if the action potential jumps down the myelinated segment, slows down, jumps, slows down, etc. And this is referred to as saltatory conduction. This action potential jumps from one node to another at the speed, or near the speed of light anyway. Even though at these nodes of Ranvier we have sodium ions rushing in and then potassium ions rushing out, in the myelinated segment we actually just have an electrical current. Instead of ions rushing in and out, we have electrons jumping from one atom to the next in the form of an electrical current. Note, in the myelinated segment, we can't have sodium rushing in and potassium rushing out because any sodium and potassium channels that are in a myelinated segment are plugged up by the myelin sheath. So what we have is a pattern of sodium and potassium rushing in and out, which kicks off an electrical current in the myelinated segment. That electrical current goes near the speed of light. The ions, even though the electrons can move at near the speed of light, ions cannot. Therefore, when we have the ionic movement in a node of Ranvier, we're going relatively slow. But then we kick off an electrical current that goes near the speed of light in the next myelinated segment. So the action potential is actively regenerated at each node of Ranvier, but passively conducted along a myelinated segment. By the way, there is a video on the learning management system in the course shell from which this still was taken, and I highly encourage you to watch that video maybe several times just to make sure you understand it. And what the video will do is compare the conduction of an action potential in a myelinated neuron to the conduction of an action potential in an unmyelinated neuron. And one thing you'll see is that it's much faster in the it's much faster in the myelinated neuron than it is in the unmyelinated one. And not only is it faster in a myelinated neuron, but it's also more efficient. Remember those sodium-potassium pumps we discussed? Remember, there's always some leakage of sodium into the cell and potassium out of the cell. In order to compensate for that leakage, we have the sodium-potassium pumps. However, there is no leakage in the myelinated segments, which means in the myelinated segments you don't need a sodium-potassium pump. Since the sodium-potassium pump requires energy, less need for sodium-potassium pumps means less need for energy. So a myelinated neuron not only conducts an action potential faster, but it does so using less energy compared to an unmyelinated neuron. Now, back to the two laws of the action potential. There's the all or none law and the rate law. The all or none law is related to the excitation threshold. If you reach excitation threshold, then you get an action potential. If you don't reach excitation threshold, you get no action potential. Not only that, but when you do get an action potential, it's always going to be of the same size. It's always going to go up to approximately positive 40 millivolts. There's no such thing as a weak action potential or a strong action potential. They're always the same size. hence the all or none law. You either get all the action potential or you get none of the action potential, nothing in between. But this raises a question: how do neurons code things like, say, the intensity of a stimulus? Let's say a bright light versus a dim light. A dim light is going to activate visual cortex neurons differently than a bright light is. But it can't do that through the intensity of the action potential, because the intensity of the action potential is always the same. So instead, we have the rate law, which says that the dim light will be coded by fewer number of action potentials, whereas a bright light would be indicated by a greater number of action potentials. Action potentials are usually measured in the number that occur within a second of time. So a dim light may cause 10 action potentials within 1 second, whereas a bright light might cause 100 action potentials within 1 second. So events are coded by the firing rate, which is the rate law. They can't be coded by the amplitude or the intensity of the action potential, because that never changes due to the all or none law. Now that we understand the property of saltatory conduction and how that is generated by oligodendrocytes with their myelin sheaths and the nodes of Ranvier in between those sheaths, we can understand a disease like multiple sclerosis. Sclerosis refers to hardening. And in the case of multiple sclerosis, we get hardening of the myelin sheaths. They become calcified and therefore less supple. And those myelin sheaths, when they become hardened or calcified, they cannot wrap as tightly around the axon, which means that we lose some of this saltatory conduction property. Namely, if you have a sclerotic myelin sheath, you don't conduct the action potential at the speed of light. It's a much slower process. So we can see two basic effects of multiple sclerosis are crossed wires. So much as if you had two electrical wires running next to each other and they didn't have insulation around them, their signals would get crossed. This can happen in axons. If two axons are running close to each other, their electrical signals can get crossed if they're not well myelinated. So we get crossed wires, but we also get, as I said, the reduced speed of the action potential. And this reduced speed can be seen with electroencephalography, a brain measurement technique that records the electrical activity of the brain. And basically the way this is done, if you're a neurologist and you have a patient you suspect might have multiple sclerosis, one way of diagnosing that is to put an EEG cap on the patient and show them visual stimuli. Now we know from previous research that a visual signal can show up in the occipital lobe. This is where the visual cortex is. The visual cortex can show up or the visual signal can show up in this visual cortex in the occipital lobe as early as 50 milliseconds. So very quickly. That's in well myelinated axons. But if the axons have become sclerotic, or more specifically, if the myelin sheaths have become sclerotic, then you lose that insulation, you lose that saltatory conduction, and therefore the conduction of the action potential is slower. So instead of taking 50 milliseconds for the visual signal to get to the visual cortex, it may take more like 300 milliseconds. And as a neurologist, if you see that it's taking 300 milliseconds for the visual signal to get back to the occipital lobe, then you've got strong evidence at that point that this person has a breakdown of the myelin sheaths along the axons, and this is usually attributable to multiple sclerosis. So thus far, we've talked about the axon and the action potential and saltatory conduction. Let's lay out a few more terms here that we'll need as we move forward. Specifically, we're going to go from talking about the action potential being conducted down this axon to talking about what happens in the axon terminal, and more specifically in the terminal button. And what's going to happen, of course, is the terminal button, when the action potential arrives, the terminal button is going to release neurotransmitter. That neurotransmitter will diffuse across the synaptic cleft. It will bind to receptors in the dendrites of the postsynaptic cell and create either excitation or inhibition. So I've introduced a few new terms here. One that we haven't talked much about is the synapse. And synapses can be of three types. They can be axodendritic, as we see here, where an axon synapses with the dendrite. But they can also be axosomatic, where the axon will synapse with a cell body or the soma. And sometimes axons even synapse with other axons, in which case we have axo-axonic connections. By far the most common synapse in the brain is the axodendritic synapse, and that's the one that we're usually talking about. That space in between the axon and the dendrite is called the synaptic cleft. As we said, Santiago Ramon y Cajal was the one who hypothesized that there was a synaptic cleft, even though he couldn't see that cleft with his relatively low-powered microscopes. It wasn't until Sanford Pele came along later with an electron microscope before we were able to actually see the space or the synaptic cleft in between the axon and the dendrites. So the cell that comes before the synaptic cleft is referred to as the presynaptic cell, and the cell after that synaptic cleft is referred to as the postsynaptic cell. As we said, neurotransmitters such as dopamine or serotonin or glutamate or GABA, these kind of neurotransmitters will be released from the axon terminal buttons and diffuse across the synaptic cleft to bind to receptors on the dendrites of the postsynaptic cell. So this covers some terminology that we're getting ready to use as we talk about this process of the action potential arriving at the terminal button, it causing the release of neurotransmitter, that transmitter floating across the synaptic cleft to bind to receptors in the postsynaptic cell. These terms will all be relevant as we move forward here.